Hello, can you guys hear me?
Can hear
Yep.
you.
Great. Sorry for being late. I lost track of the time here and didn't realize it was after 5. All right, before we get started, are there any questions? No questions. All right, let's get started. We need to finish up the last chapter we were working on. I think it's chapter 13. And we need to finish our last chapter, which is chapter 7. Is that right? I don't seem to have chapter 7 on here, so I'll have to get my thumb drive. Yep. So we should be finished with chapter 13. We're almost finished with it. There is no lab today or any of this week. Today is June 7th. Please note that your unknown project is due Saturday, June 11th. The unknown notes pages will close Friday, June 10th. Any questions about any of that? Where's my, there it is.
So for the unknown, we just turned in the whole unknown notes page as our unknown report plus a paragraph at the end.
No, your unknown report must be a formal report.
Okay.
If you did the unknown notes page formally, and I don't think many people did, at most one student did, it would be very similar, but most of you were very relaxed with your unknown notes, and that's fine. So there's an example. Let me see if I can call that up. There's an example on the web page for doing the unknown report. So make
Okay.
your report look something like that example.
Okay, thank you.
Okay. I'm trying to get there. Actually, I don't think I'm showing my screen yet, am I? Let me go ahead and do that.
I have a question, Dr. Volpe.
Just a minute. I'm trying to show you what the formal report should look like. You should be seeing my screen now. So the sample journal gives an example of the unknown report. Do not have a title page as the unknown report example has. I should try and edit this, although I don't think I have this in a Word document. So in your unknown report, you should have a day, and then the observations you made on this day, and that will be generally from tests that you ran earlier because you requested them earlier. And then you maybe got your results on February 6th. And so state what observations you did, state what tests you requested on this day, and then you should state why you requested them. And at the end, you should have what your conclusions are. And the conclusions are just the interpretations of your results. Let me go to another example where it'd be a little easier. All right, so the observation was the mannitol broth. The broth was a bright yellow. The conclusion would be acid was grown in the, or acid formed in the mannitol broth, and there was no gas bubble, so no gas was produced. Okay, any question about that? So the observations are your results. The conclusions are your interpretations, what the results mean. All right. And in this day, the instructor put the conclusions and activities together. You don't have to have them separate, but it's probably best to just do it all the same. And the activity is he wants to inoculate a 7% sodium chloride plate. And then the reason is he has either Staphylococcus aureus or Micrococcus luteus. And Staphylococcus aureus should not grow on sodium chloride plates. Actually, I'm not sure the Micrococcus luteus does not grow. It might not have grown here. Let me take a look at that and make sure that that's the case. This will take a little while to download. You're not going to see this very good because I'm just going to use this page here. There we go, 7%. And what column is that? That one here. All right, on our chart, Micrococcus luteus does not grow at 7% sodium chloride. So the instructor was correct. All right, any questions about any of that? Any other questions?
Did you say not to make a title page like the one in the example?
You should not make a title page. You should have your title information on the first page of your text, meaning don't make it a separate page. Okay, you should have the title information, but just stick it all together on the first page. So all of this information, see if I can get that.
Just like put it at the top.
Yeah, just put it at the top of this page here.
Okay, perfect.
Okay.
And Dr. Volpe, is there a limit to how many pages we should have, or it depends on our tests?
There is a limit. I don't remember what it is, but you can find the instructions. Okay. And
Okay,
there's a
and
simple way to get around that limit. If you're too big, you can single space the sentences. I'm sorry, I didn't hear that. All right, maybe that wasn't a question. Somebody was talking there in the background. There is a limit. I don't remember what it is. Generally speaking, as long as you're not a page over the limit, you will not be penalized. Okay. And there's a limit on how small it can be. I don't remember what that is either. It's something like 5 pages. And if you're more than a page shorter than that, you will get a penalty. Okay. And there's an easy way to expand that. You can use a large font. You can double space the typing. Okay. Any question about any of that? But yeah, it has limits, and look up on the instructions for what the limit exactly is. And the instructions tell you how to do the report. So you should look at that before you write the report. All right, let me get chapter 7 copied.
I have one more question. When will our infectious disease project be graded?
Well, before I have to turn in the grade, of course. But I can't tell you exactly when that will be. Hopefully it'll be graded within a week after you turn it in. All right. Now, as you know, your infectious disease project, I have not finished grading that. So I'm trying to get there. I'm a little behind on the grading. Let me go put this here now. All right, so any other questions? All right, let's find where we were. Let me remove the thumb drive here. So we'll finish chapter 13 and start chapter 7. I do hope to finish chapter 7 so that next Thursday we'll only have review. But I do have some catch up in case we don't finish chapter 7. Let me close that. And eject that and remove my thumb drive so I can put my headphones back on.
Did we finish? Are you finishing chapter 13 or chapters? Are we starting chapter 7 today?
We're finishing chapter 13.
Okay, thank you.
I think we're beyond that. There we are. We need to talk a little bit more about prions. So this is where we left off. Remember, prions are an infectious proteinaceous particle. So it's a protein, infectious proteins. They're inherited, so they can be passed from parents to offspring. They are transmissible by ingestion, by transplant, like if somebody has a prion disease and then they take the organs from the donor and put them into a recipient, the recipient can come down with the prion disease, and they can be transplanted by surgical instruments or a needle. We've gone spongiform encephalopathy. These are vacuoles in the brain. And the most common case of spongiform encephalopathy is mad cow disease or bovine spongiform encephalopathy. We don't call it that in humans. It's called Creutzfeldt-Jakob disease, Gertzmann-Straussler-Schneckner syndrome, fatal familial insomnia, and Kuru. And we'll talk a little bit more about each of these. Kuru was the disease seen in the Fore tribe in Papua New Guinea. This is Papua New Guinea, and the red marks the region of the Fore tribe. Kuru was seen when the patient was unable to walk, loss of the ability to swallow or chew, drastic weight loss, and then the wasting away that led to death. How was it transmitted? Kuru was transmitted during the 4 funeral rituals where people cooked their dead relatives and then ate them. Okay, any question about that? In the funeral ritual in the Fore tribe, they actually ate their dead relatives, and that tended to spread Kuru, a prion disease. Any question about that? what happened was probably one individual came down with bovine spongiform encephalopathy in the 4 tribe, and then their dead family members, the family members ate the dead, and then that spread the kuru among the 4 tribe. Any question about that? Kuru is pretty much extinct now in the 4 tribe. For one thing, the mothers noticed that their children were getting kuru after consuming their dead relatives. So the mothers prevented the children from eating the flesh of their dead relatives, and they stopped eating it as well. So generally, by the time Western civilization made contact with the 4 tribe, the mothers and the children were no longer getting kuru, but the men continued the ritual. And then the missionaries came in and they said, eating the dead, that's horrible. We're not going to allow this. And they put a stop on the practice. Okay, so the missionaries with their oh, I don't know what you want to call it, their one belief, and their belief is best, actually did something good for the 4 tribe, because it kind of eliminated kuru. And then of course now there's laws in Papua New Guinea that you no longer eat the dead, and the 4 tribe pretty much has no more kuru. Any question about any of that? I've already showed this picture. Seeing the brains, there's a normal brain, what it normally looks like. And then the spongiform disease where the brain looks like it's a sponge. How the protein spread was, there is a prion form of the protein and a normal form of the protein. The normal form of the protein, which is PRPC, is a normal protein on the cell surface of nerve cells. So it is necessary for the functioning of nerve cells. Any question about that? And then the prion form of the protein, which is PRP sub, or what do you call that? Superscript SC, is the prion form of the protein, and it has the same amino acid as the PRP subscript or superscript C, meaning the normal form of the protein. So it is the same protein. The only difference is the folding pattern of the protein. The prion protein is folded in a different pattern than the normal protein is folded. And how the prion protein replicates is the prion protein comes up to the normal protein and then causes the normal protein to change its folding pattern into the prion form of the protein. Any question about any of that? Oh, SC stands for the Scrapes protein. Scrapes is a prion disease that occurs in sheep. Now, the nerve cell realizes there's not enough of the normal PRPC protein, so the nerve cell makes more of this protein. The trouble is this cell now has 2 prion proteins, and either one of these can come up to the new protein and convert it to the prion protein. And so the cell makes more of the normal protein, and then the prion form of the protein makes more of the prion protein. And that's how the disease manifests itself where the prion protein, I don't know, copies itself or replicates itself. The abnormal prion protein accumulates in the brain cells and then leads to damage of the cells, forming the plaques in the cells and in the brain, and eventually killing the brain cell, meaning it leads to cell death. Any question about any of that? All right. The worst case of prion disease was mad cow disease in England. The English had an atrocious habit where they wanted to sell the brains of the cows, but nobody wanted them. So they ground up the brains of the cows and then put it on grain and then fed the grain to the cows. Well, a prion infected cow had this happen, and its brain was ground up and then fed. Its brain was ground up and put on the grain, and then the grain was fed to cows, and that led to more cows getting the prion disease. And that led to mad cow disease, an epidemic in Great Britain, which affected, obviously, cows, people, and cats. There were about 150 British who came down with Creutzfeldt-Jakob disease, which they think was obtained by eating prion contaminated beef. All of the individuals who became prion infected had been eating beef, okay? And the cats, they know, were eating prion tainted beef as well, meaning just cat food that had beef in it. Any question about that? So 150 British cats and about 150 British individuals came down with prion disease, most likely from eating contaminated beef, because the beef in England at the time was potentially contaminated with prions. And of course, the cows, there was thousands of cattle, maybe even more, hundreds of thousands of cattle that came down with prion disease. Any question about any of that? Any question about prion disease? If not, let's briefly talk about plant viruses and viroids. Plant viruses resemble animal viruses in many ways because many diseases that are economically important to crops, such as bean, corn, sugarcane, and potatoes, they do not infect and do damage to humans, but they damage our crops. Because of the tough cell wall, plant viruses tend to enter through wounds in the plant cell or via a parasite like a nematode, a fungus, or an insect. And then infected plants can spread the virus in pollen or seeds, as well as an insect can land on the infected plant and then carry the disease to another plant. Any question about plant viruses? We're not going to talk about plant viruses because they don't infect humans, but that's all I wanted to say. Viroids are naked RNA molecules. They have no protein coat, but you can think of them as a RNA virus without the protein coat. Viroids are naked RNA because diseases in plants. There's the potato spindler tuber viroid, which is an RNA when it infects the potato plant, causes the plant to make smaller potatoes. What's called spindle tuber viroid. There are no viroids known that infect animal cells. So no viroids infect humans. Any question about any of that? And the last slide is not important, so don't remember this, but they found some very large viruses that infect amoebas. And the very largest virus is actually a little larger than the smallest prokaryotic cell, the Pandora virus, the very largest virus. Any question about any of that? All right, any question about viruses, viroids, and prions? If not, let's close that down. And open up chapter 7. So chapter 7 is the control of microbial growth, things like disinfectants and antibiotics. Not antibiotics, antibiotics is a part, I'm thinking of disinfectants and antiseptics, okay? So students generally have an easy understanding of this because we all know what disinfectants are and we know how they work. So I move this chapter to the very end because it's this chapter that you already have some good understanding of. I know the terms, know what sterilization is, disinfection, antisepsis, degerming, sanitization, autoclaving, pasteurization, what's a biocide, what's a germicide. Be able to discuss the factors that influence microbial death rates, be able to discuss the 3 main ways in which antimicrobial agents kill or inhibit cells, understand the various types of disinfectants and how they work, and then understand the relative resistance of the major microbial groups to antimicrobial agents. And we'll talk about that at the very end. Any question about anything we're going to do in this chapter? All right. The rate of microbial death. Bacterial populations die at a constant logarithmic rate, however you kill the cells, like when heated or treated with an antimicrobial chemical. So on a log scale of the number of cells versus time, which is an arithmetic scale, we note that the population of cells dies constantly over time, or a straight line. Generally, over 1 unit of time, you get 1 magnitude of cell death, or 90% of the cells die over 1 minute of time. So if you have 10 to the 6th cells, 1 minute later, you'll have 10 to the 5th cells. 2 minutes later, you'll have 10 to the 4th cells. Any question about any of that? And this is always seen, the difference is that the death rate may be slower or it may be more rapid. Any question about that? So per unit time, you get 90% of the cells dying in 1 unit of time. So 100,000 or 1 million becomes 100,000. The effectiveness of antimicrobial treatment depends on a couple of factors. First, it depends on the number of microbes present at the beginning of the treatment. The more microbes that are present, the longer the treatment has to be in order to kill off all of the cells. Any question about that? So the number of microbes present depends on the effectiveness. So if there's 10 to the 6 cells and you want to reduce it down to 1 cell, you need to expose it for 6 minutes on this treatment. If there are fewer cells, like 10 to the 3rd, you'd only need to expose it for 3 minutes to get it down to 1 cell. There are environmental influences that can affect the effectiveness of the antimicrobial treatment. The presence of organic matter can affect the effectiveness of the treatment. Hospital specimens, you should note, usually come with organic matter. Like if a patient throws up, they'll have organic matter in their upchuck. If they have diarrhea, they'll have organic matter in their what do you want to call that? Their mess. The temperature of the treatment can affect the effectiveness of the treatment. Generally, with warmer treatment, the more effective the antimicrobial treatment will be. And with lower temperature, the less effective it will be. The presence of biofilms also affects the effectiveness of the antimicrobial treatment. If the organism makes a biofilm, you'll have a much harder time killing off the species than you would if there's no biofilm. Any question about that? As I mentioned, the time of exposure can affect the effectiveness of the antimicrobial treatment. Generally, a longer period is better for killing off all of the cells. And for spores, like endospores, you need a very long time of exposure to the treatment to kill off the organism if the treatment kills the endospores. Many antimicrobial treatments will not kill endospores. What's an example? Penicillin G will not kill endospores. The soap that you wash your hands with will not kill endospores. Alcohol will not kill endospores. And that has to do with the microbial characteristics, which I'll discuss this at the end of this lesson, but the different microbial characteristics can influence the effectiveness of the antimicrobial treatment. For example, Mycobacterium has a cell wall which has a lot of mycolic acid in it. That makes Mycobacterium resistant on many antimicrobial treatments. Any antimicrobial treatment that is using a water-soluble antiseptic or disinfectant will not work well on Mycobacterium because water-soluble agents cannot get inside the cell wall and cannot get inside the cell to kill the cell. Any question about any of that? And then I'll talk about this a little bit later. If you have a Mycobacterium, alcohol is actually good at killing the Mycobacterium. Alcohol is such a weak antiseptic or disinfectant that we don't even call it an antiseptic or a disinfectant. We just generally call it a degerming agent because it doesn't really kill all of the microbes on your skin when you apply the alcohol. It simply reduces their number because alcohol is not a very good antibacterial agent, but it is very effective on Mycobacterium because alcohol can get right through the mycolic acid of their cell wall. So there's one microbial characteristic which makes these cells very resistant to most antimicrobial treatments, but it makes the cells very sensitive to the treatment by alcohol, which is not a very effective antimicrobial agent. Any question about any of this? All right, so this slide is showing you the different terms and what they mean. Sterilization, the destruction or removal of all forms of microbial life, including endospores, but not including prions. Prions are very difficult to kill. I should have mentioned that previously when I was talking about prions. To kill prions, you need to either subject them to an enzyme that digests proteins, meaning a proteinase, or you have to submit the agent to 1,000 degrees Fahrenheit to kill the prion. And we do not know why prions are so difficult to kill. But sterilization does not refer to the removal of prions, but it does refer to the removal of endospores. Sterilization in the microbiology lab is usually done using an autoclave, using steam under pressure, although there are other ways to sterilize, like using sterilizing gas, ethylene oxide, or what's the other way? Oh, you could use a peroxide. It does kill endospores. We'll talk a little bit more about that. Commercial sterilization is applying sufficient heat to kill endospores and Clostridium botulinum in canned food. So this is more used for the industry to make canned goods. Disinfection refers to the destruction of vegetative pathogens on an object. So when we put bleach on the toilet, we're using a disinfectant, meaning an agent that works as disinfection. It kills vegetative pathogens, but it does not kill endospores. Disinfection can use a physical or a chemical agent. An antiseptic is destruction of vegetative pathogens on living tissue. It does not kill endospores. So an antiseptic agent and a disinfectant agent is only what you're applying it to. If you put bleach on a toilet or a sink, we're using it as a disinfective agent. However, if you put the bleach on the skin of a patient, it is then an antiseptic agent. Generally speaking, antiseptic agents are more mild and less toxic than disinfective agents. But we actually do use bleach to treat necrotizing fasciitis on patients. Although that could be an older treatment. I don't know if they still use it. But initially that was how they treated necrotizing fasciitis on patients. Antiseptic agents is almost always by a chemical antimicrobial agent. And a good antiseptic agent would be tincture of iodine, which they used to use very often in the hospital. They don't use it as much anymore. It's still used, but not as much. But degerming is removal of microbes from a limited area, such as the skin around an injection site, or a site that you're going to degerm before you give an injection. It's mostly a mechanical removal with an alcohol-soaked swab. Degerming.
I have a question, Dr. Volpe.
Huh?
Would that be degerming when betadine is used on skin?
Well, betadine is a little bit stronger agent, so they usually call it an antiseptic. Like, don't they use betadine before surgery?
Yeah.
Yeah, that would be an antiseptic agent. It's a little bit stronger and it kills off more cells than a degerming agent. Okay,
Thank you.
all right. Sanitization treatment intended to lower microbial counts on usually eating and drinking utensils to lower the microbes to safe public health levels. So you can think of your dishwasher as sanitizing your dishes. But they do disinfect the dishes and what do you call that? The food industry and a chemical disinfectant. A biocide or a germicide is a substance that kills antimicrobial agents. So it kills microbes. Oops. Oh, there it was. A biocide or germicide does not usually kill endospores. We have different names for certain ones, like bactericides is an agent that kills bacteria. Fungicides are agents that kill fungi. Viricides, they inactivate viruses, but all of them together are a biocide or a germicide. Bacteriostasis is when you inhibit the growth of microbes, but you do not kill the microbe. And we talked about this. A bacteriostatic antibiotic is one that prevents the microbes from growing, and it allows your immune system to then kill off the microbe, because the bacteriostatic antibiotic does not. When you remove the bacteriostatic agent, the growth may resume. If it hasn't been killed off by the host immune system, the bacteria will resume once you remove the bacteriostatic agent. All right, any question about any of this? Sepsis refers to microbial contamination. Asepsis is the absence of contamination by unwanted organisms. So aseptic technique is what you learned in microbiology, and it is important in surgery to minimize the contamination from the surgery, especially from the surgical instruments. But you also use aseptic to prevent contamination from the operating personnel, from the patient themselves, and from the air. And so nowadays we usually have surgery done under aseptic conditions where the surgeon wears a mask and headgear to prevent microbes from the surgeon infecting the patient. And then the instrument is sterilized and they wear gloves. All right, any question about any of this? Antimicrobial agents kill or inhibit microbes by three means. They can alter the membrane permeability of the microbe. They can damage the intracellular proteins of the microbe, or they can damage the nucleic acid of the microbes. If you damage the plasma membrane, either the lipids or the proteins of the membrane, it will put a hole in the membrane, causing the cellular contents to escape the cell and interfere with normal metabolism, resulting in the death of the cell. An example of an antimicrobial agent that puts a hole in the cell membrane is polymyxin B. That antibiotic puts a hole in the plasma membrane. Any question about any of that? You can also damage intracellular proteins. You denature proteins, especially enzymes, by either targeting disulfide bridges or just causing the protein to unfold, and then you can kill the cell. This is actually one of the ways which ethanol damages cells. It denatures proteins. Of course, ethanol can also damage the membrane, the cell membrane. Tetracycline is an antibiotic which damages the microbes' proteins. And so that would be one that only damages the proteins. Any question about that? All right. You can also damage the cell by damaging their nucleic acids. If you damage the nucleic acid enough, and that's usually a little more than 5 or 6 times, it often results in cell death. You only need to mutate one critical gene, and that will kill the cell. An example of damaging nucleic acids is subjecting the cells to ionizing radiation like X-rays. You're damaging the nucleic acid, which kills cells. And as you know, we actually sterilize some food that way. And if you ever see a meat product on the shelf in the grocery store that's been sealed, it's probably been treated with ionizing radiation. And that's why the meat does not require cold temperatures and will not spoil. There are other ways to do it too, but that's often the way it's done. Any question about any of that? All right, let's talk about the physical methods for controlling microbial growth. You can heat microbes, you can filter them out, you can use a low temperature to control their growth, and this doesn't tend to kill them, it tends to stop their growth. You can use high pressure to kill them, you can use desiccation, you can use osmotic pressure, and you can use radiation to control microbial growth. So heat's the first physical method we're going to talk about for controlling microbes. All methods of heating kills microorganisms by denaturing proteins and enzymes. However, flaming and incineration, which are high temperature heat, can actually turn cells into char. And that, of course, will damage the cells as well. Wet heat is boiling or autoclaving or steaming, and it's a little more effective than dry heat, which is baking. Obviously, flaming and incineration are a very high temperature heat, and they are very effective at killing cells in a short period of time, but this is very high heat. So for the same temperature, we'll talk about this a little bit more. The same temperature, wet heat is more effective than dry heat. And the reason is moist heat denatures, coagulates proteins and enzymes. You just need to boil something like at 100 degrees Celsius, or put the cells in flowing steam, and that kills vegetative pathogens, most viruses, fungi, fungal spores, within about 10 minutes. So if you cook your vegetables for 10 minutes, you'll kill off all of these agents. Oftentimes, the cells will be killed off much faster. Like you only need to subject E. coli to 100 degrees Celsius boiling temperature, and then that kills the E. coli. And we're talking about a second or less. So oftentimes you can kill the cells much faster than 10 minutes. Note that boiling does not kill endospores. Endospores and some viruses take longer to destroy. Some endospores can survive greater than 20 hours in boiling water. So some endospores are very resistant to heat, moist heat, as well as most other agents you use to treat the microbes. So reliable sterilization using moist heat requires temperature above boiling. How is this achieved? By autoclaving. Autoclaving is a very large pressure cooker where it's very effective at killing cells because you have the cells exposed to a high temperature. It's usually 121 degrees Celsius or higher under pressure, and that's 15 pounds per square inch. And then that kills most microbes within 15 minutes. It does not kill endospores, but it does kill most microbes, and it does kill some endospores. Okay. And in 15 minutes, it actually kills all organisms. And after 15 minutes, it kills endospores if we're talking about a liter in size. If it's greater than a liter, you need more than 15 minutes. All right, any questions about an autoclave? Pasteurization is something that Louis Pasteur invented in 1857. It does not kill all microbes, but it does reduce the number of spoilage organisms, killing all pathogens. It is mild heating, sufficient to kill organisms that cause spoilage. It denatures proteins and enzymes. However, thermoduric organisms can survive under the pasteurization. Thermoduric organisms are heat resistant. They might not grow at this temperature, but they can survive it. There are two methods to pasteurize milk and most other agents that are pasteurized. You can expose the agent or the milk to 63 degrees Celsius for 30 minutes or to 72 degrees Celsius for 15 seconds. Most milk that is pasteurized today is treated at 72 degrees Celsius for 15 seconds. And that allows the milk to last longer than it would if it was unpasteurized. However, it still has a shelf life because it does not kill all microbes in the milk. And those other microbes will grow over time and spoil the milk. Milk can be sterilized, and this is not pasteurization. It's called ultra high temperature or UHT milk. And the milk is exposed to 140 degrees Celsius for 1 second, which sterilizes the milk from everything except for endospores and prions. This milk does not require refrigeration until you open it. And you can sometimes find this milk on the store at room temperature. Any question about any of that? All right, let's talk about dry heat. Dry heat kills things by sterilization or killing by oxidation. Examples being flaming and incineration, which is very high temperature, flaming and incineration, and it happens very quickly. Hot air sterilization, such as baking at 100 degrees Celsius in your oven, requires exposure for 2 hours to be effective, meaning dry heat sterilization is not as effective as wet heat sterilization, wherever I've got that, which at 100 degrees Celsius, you only need 10 minutes. And this cat is trying to show you that dry heat is not very effective on killing microbes, meaning the cat climbed into the oven because it likes the warm oven. And you all know that dry heat is not as effective as wet heat. For example, when we take stuff out of the oven, we are exposing our hands to that oven temperature. When our hands are fine, because it's a limited exposure to the heat. But you cannot put your hand through steam because you'll get burned instantaneously if you put your hand through steam. And once again, that's showing you the wet heat is more efficient at killing than dry heat. So wet heat is more efficient in regard to time than dry heat. Any question about any of that? Filtration removes microbes from liquids and gases. You have pores in the filter, and then you pour what you want filtered, and then send it through the membrane. There's the membrane there. All the microbes will be on this side of the membrane, and then the solution down here will be microbe-free, at least when we're talking about bacteria. You can have filters small enough to remove viruses, but those are very tiny pores. We use filtration for sterilizing liquids that would otherwise be altered by heating. Like some examples includes juice, where we sterilize it. Because if you were to heat it, you would have a different flavor. You would destroy the vitamins if you were to heat it. And you might change the taste. A Coors beer is actually a beer which is filtered. It is not pasteurized. Vaccines are sometimes filtered. Milk is usually not filtered because the milk lipids, as well as the milk proteins, would clog the pore, and then the filtration would stop. So milk is not usually filtered. It is usually pasteurized. All right, any question about any of this? Okay, low temperature can be used to control microbial growth. Low temperature generally inhibits microbial growth. It generally does not kill cells. The cells would be very finicky if the low temperature killed them. Refrigeration at temperatures between 0 and 7 degrees Celsius slows the metabolic rate of microbes so they cannot reproduce or synthesize toxins. But do understand that psychotrophs can grow and reproduce at these temperatures. And they would grow or replicate 3 times per day. And after about a week, we'd have about 2 million cells, which could cause food spoilage. So when you refrigerate things, you do not necessarily keep the food so that it can stay forever. All it does is it gives you a longer window of time. Like milk can be refrigerated at least 1 week after opening. Meat, after you cook it and then you put it in the refrigerator, it can usually last 1 week in the refrigerator. Most things can last 1 week in the refrigerator, but there are some things like ketchup, which can last much longer. And that's because besides the temperature, ketchup has a low pH and a high salinity, which keeps microbes from growing. Freezing does stop metabolic activity. It does not kill all cells. It does kill, I don't know, about 50% of bacteria, but not all cells. So freezing does not sterilize like food. Slow freezing in your home freezer allows ice crystals to form, which do kill some bacteria, but not all bacteria. But the slow freezing does kill all eukaryotic parasites. So any parasitic organism would be killed by slow freezing. And slow freezing is normal freezing in your home freezer. If you were to quick freeze, like putting the bacteria in liquid nitrogen, it would freeze so quickly that ice crystals would not form, and that would not kill bacteria, and it may not kill eukaryotic parasites. Of course, I don't think most of you have the ability to freeze something with liquid nitrogen. You can actually make ice cream by putting the ice cream agents in the liquid nitrogen. High pressure is another method that can be used to control microbial growth. High pressure transfers the pressure evenly throughout a liquid, and this kills vegetative bacteria cells. High pressure denatures proteins and alters carbohydrates. Endospores are not killed by high pressure. But high pressure does preserve the color, the flavor, and the nutrient value of the agent being subjected to the high pressure. And that is why juice is frequently killed or treated with high pressure before it's sold. Most juice is not filtered. It's usually treated with high pressure, or it's even treated with boiling, in which case you do destroy the nutrient value, at least vitamin C. Desiccation and osmotic pressure is another way of controlling microbial growth. Desiccation is the drying. This prevents microbial metabolism. Some cells can remain viable for years under desiccation, like endospores can remain viable. Mycolic acid can remain viable for months, meaning a microbacterium that has mycolic acid in it can remain viable for months under desiccation. The resistance to desiccation varies species by species. E. coli would be very susceptible to death by desiccation. Some examples where we use desiccation mean when we dry fruit, such as dried figs. Example of desiccation to keep the food from spoiling. So dried fruit, such as figs. And humans are being used in desiccation to prevent food spoilage for, well, I would say probably 4 millennia. Osmotic pressure is another way we can control microbial growth. High osmotic pressure, like high salt and high sugar, causes plasmolysis, meaning the water will leave the cell and then the cell membrane will pull away from the cell wall, causing plasmolysis. And for many bacteria like E. coli, this will kill the cells. Osmotic pressure and desiccation do not kill endospores. All right, any question about desiccation on osmotic pressure? If not, let's move to the physical methods for controlling microbial growth. The physical methods tend to use radiation. Radiation damages DNA. There's 2 types of radiation: ionizing radiation, and then UV light, which is called non-ionizing radiation. Ionizing radiation involves X-rays, gamma rays, and electron beams. They have a short wavelength. They ionize molecules in water, producing ions and hydroxyl radicals. These molecules then react with other organic molecules, especially DNA, and then damage the DNA. So this chart is trying to show you that actually all of the electromagnetic spectrum is radiation, but when we're talking about ionizing radiation, we mean here it is there, gamma rays, X-rays for ionizing radiation. They hit molecules and atoms, giving the molecule and atom enough energy for the electron to leave that molecule, creating an ion. And then that ion can bump into a region of the cell, damaging it, such as DNA. Any question about any of that? All right, the next form of radiation we need to talk about is non-ionizing radiation. There's only really one non-ionizing radiation, and that's ultraviolet light or UV light. UV light, when it hits DNA, causes damage to the DNA in that the 2 thymines that are next to each other get a thymine dimer from the UV light. And that thymine dimer creates a bubble in the DNA, and this bubble cannot be transcribed or cannot replicate. And so if the cell is trying to replicate, it can fix this mutation, and then there's no way to go back. But once the DNA is damaged, the cell may notice the damage, the thymine dimers, and then engage in DNA repair to remove the damage. Any question about any of that? I guess we're here. Microwave radiation is not particularly antimicrobial inside an object. It is effective in treating surfaces or the outside of a microbe or some object. And it depends on, I guess, the type of radiation, like gamma rays can penetrate the object, but UV light cannot. So I guess what I'm trying to talk to you is that UV light is only good on killing surfaces and the air. It's not good at killing things inside of an object. All right, these tables show you a summary of the different methods for controlling microbial growth. And we've talked about each of them. There's moist heat, which is boiling or steam, autoclaving. And these are good at effectively killing off vegetative pathogens, but they are not so good at killing endospores. Although endospores will die under an autoclave. Pasteurization is a form of moist heat, which we do not use to even degerm something. We use it to simply make it safe to eat and then increase the shelf life. Dry heat is effective if we're talking about flaming or incineration, but these are very high temperatures. For other temperatures, hot air sterilization for baking in an oven, it's not very effective. You need 2 hours of time if you're only subjecting the object to 170 degrees Celsius. But it is effective at killing vegetative cells. And so they will frequently sterilize instruments with hot air sterilization. And to tell you the truth, I do not know why they don't use autoclaving as opposed to baking in the hospital. My guess is that they have lots of ovens in the hospital, but they either don't have an autoclave or they only have one. Filtration is very good at removing bacteria from a liquid or a gas. You just send the agent through a filter. And then what comes through the filter has no bacteria. Or for that matter, no eukaryotic cells, such as parasites. All right, cold is another mechanism of controlling microbial growth. Cold acts more as a bacteriostatic agent, meaning the microbes typically do not die in the refrigerator. High pressure, another way we can control microbial growth. Apply pressure to an object, and that preserves the color, the flavors, and the nutrient values. And that's a way that a lot of fruit juices are sterilized, not sterilized, but biocided. Desiccation, this involves the removal of water from microbes. So it's primarily bacteriostatic, although to organisms that do not like undergoing their water loss, like E. coli, it would kill cells. Desiccation is used in food preservation. I forgot to give the examples. Salting fish would be a good example of how we use desiccation. Drying fruit, like dried pineapple rings, and we're using desiccation to help prevent the growth of the bacteria. Osmotic pressure, similar to desiccation, results in loss of water from microbial cells. And that would be, once again, the salting of fish or the drying of fruit. And when you're drying fruit, you have high osmotic pressure because the sugar content increases in value in the fruit, making it so bacteria cannot grow on the dried fruit. And then radiation, ionizing and non-ionizing radiation both damage DNA, although they do it differently. Ionizing radiation causes an electron to leave a molecule, and then that molecule can bump into DNA, damaging the DNA, or the ionizing radiation can hit the DNA and cause the DNA itself to lose the electron. Non-ionizing radiation is UV light, where it damages DNA by causing thymine dimers in adjacent thymines. So the thymines have to be adjacent to each other. All right, any question about any of that? If not, I'm going to stop here and I will continue with this next Thursday.
I do have a question.
Sure,
Could you upload the chapter 13 PowerPoint?
sure.
And then I'm also not finding the lecture video from last Thursday on YouTube.
That was uploaded. So refresh your browser and then look again.
Okay, I just checked it.
It's there, it should be there. Let me
It's
check
so,
it myself.
yeah,
'Cause
what
I
kind, what,
uploaded it. I didn't check to see if it finished its, what do you call that? Because YouTube does some changes to what I upload too. So I didn't check that. but
gotcha, because
it's there.
right now what I'm seeing is the one from May 31st is on there twice.
Oh,
So I don't know if maybe it like,
maybe I did a mistake and I did that one twice. Let me check.
okay, thank you.
Okay. All right. Anything else?
Yes, do we have like a study guide for the final, or do we just use these study guides to study from?
You should use your notes to study. However, oops, I'm not doing this right. However, I do have the oh, what is it called? The chapter objectives. So you can take a look at the chapter objectives to each chapter and then use that to study for the final. And then on Thursday, I will go over material for the final.
Thank you.
All right. All right. Any other questions? See, that should be there. And what is that? I can't see what it says. No comments. All right, I'll figure this out. I'm not seeing it, but I'll figure it out. Any other questions or comments? Oh, there it is there. So that's from 531. And that one is from 531. Yep, that's there twice. All right, I'll try and get that corrected.
Thank you.
All right, bye.
Thank you.